Welcome to part two of a three-part series on microbial pathogenicity. In the first part, I gave a brief introduction to pathogenicity, and the predominant part of the discussion was biofilms and how biofilms increase pathogenicity, especially of virulent strains of bacteria. So now we'll look at some more details, specifically focusing in on bacterial pathogenicity. What are some structures that they possess that increases their pathogenicity? Well, many bacteria produce a capsule, and capsules are kind of think of it like a suit of armor that surrounds the bacterial cell wall. And because of this shield, so to speak, that's around the bacterial cells, this leads to impaired phagocytosis. And so when a phagocyte engulfs a encapsulated bacterium, phagocytosis is ineffective because the capsule is resistant to the enzymes that are within the lysosomes. And so therefore they resist it, they impair phagocytosis, and so this then as a result increases the virulence of that particular bacterial strain. So in some way, one way they can evade phagocytosis, they can neutralize or the enzymes are ineffective against the bacterium because of the capsule that it produces. Something else that bacteria possess are siderophores, which are compounds that take iron from the host. And so the affinity for iron to a siderophore there's a strong affinity between the two of them. And so therefore the bacteria, if they possess enough of the siderophores or the correct siderophore, if whatever environment it happens to be living in, say if this is a pathogenic bacterium that lives in the blood, it can take iron from the blood and then it can thrive as a result of that because it now has the iron available for it to carry out all of its metabolic functions. Antigenic variation. So this is when surface antigens alter, sometimes called genetic variation or genetic drift or genetic shift in some ways. So those terms are related to this idea of the surface antigens undergoing variation in some form or another. And so as a result of this change of antigens, antibodies then cannot attach to the antigens that are on the surface of the bacteria. Now, on the antigens, there's an even smaller component called an epitope. And the epitope is actually what the antibodies will bond to. And so if those little tiny epitopes change ever so slightly, that creates a problem because now you have to go through a whole cycle of making new antibodies to fight against that particular protein, carbohydrate, lipid compound. And so as a result of this variation, you develop resistance against that particular bacterium, but then it undergoes variation, and now you have to go through the whole primary response system where you have to make a whole new round and a whole new set of antibodies, which I will talk about in another lecture on specific immunity. So anyway, bacteria undergo antigenic variation, as do viruses, as do protozoa as well. They vary their antigens, and as a result, antibodies are ineffective. The M protein. The M protein is on the cell wall of bacteria, and it is a heat and acid resistant protein, and so it makes sense. So it can resist high temperatures, it can resist stomach acids, for example, or if you're using an acid-based compound, for example, to try to eliminate the bacteria, these M proteins are enable the bacteria to be resistant to those conditions. A protein called OPA, it's short for opaque because of the nature of the protein being an opaque protein. It enhances adherence, and so it enables them to not only stick to a surface, such as a mucous membrane or microvilli in the intestinal wall, but it also enables them to adhere better to each other as well, to create a biofilm and to enlarge a biofilm, and so it can increase pathogenicity that way. But also at the same time, this OPA protein can enable them to move their way through a tissue as well, through through layers of cells. So this is just an adherence protein that enhances the adherence ability of the bacterial cells. Waxy lipids, they're within the cell wall. They resist phagocytosis, and tuberculosis, the tuberculosis bacterium, is notorious for this, and this is what enables it to be so pathogenic, to form the tubercles that they form within, say, our lungs, for example, especially there, but in other parts of the body as well. The waxy lipids then prevent phagocytosis because the enzymes cannot break down these lipids, and so the bacteria then can survive and live. They have invasins, which are, again, proteins. Everything seems to be based on proteins, and of course it would be, because that's what the genetic code is there for, to lead to the production of proteins. They rearrange the host cell's cytoskeleton, and that process, through the rearrangement of the actin filaments and the microtubules and the intermediate filaments, that's called membrane ruffling. And what that does then, it makes invasion into the host cell easier. And you see why they're called invasins, because invasion is easier for the bacterium to then kind of work its way, weasel its way into the host cell and then do its damage, parasitizing the host cell, for example. Some enzymes that they produce. Coagulases will lead to the coagulation of blood, and bacteria, many of them are classified as coagulase positive and coagulase negative. The ability to coagulate blood, well, you say, well, what's the benefit of that? Well, within within the blood clot, the bacteria then can multiply and divide, and so they have these enzymes that can do that. Kinases are enzymes that break down fibrin clots, so that if they do happen to replicate in there, they can then break the clot and then travel through the circulatory system that way. If the body is trying to create a clot to isolate the bacteria then can digest the clot, break it down, and then be able to disseminate more quickly and easily through the use of this enzyme. Hyaluronidase is an enzyme that breaks down, hydrolyzes hyaluronic acid. One that's notorious for this would be the flesh-eating bacterium, group A Streptococci. They have hyaluronidase, and they can break down hyaluronic acid in the connective tissue. That can then lead to cellulitis, and if let go, and in extreme cases, of course, you have necrotic tissue and necrotizing fasciitis. Collagenase hydrolyzes collagen, so it's kind of doing the same thing there as hyaluronidase in the connective tissue. Breaking down these proteins and utilizing their constituents for themselves and getting energy from that. IgA proteases, enzymes that destroy IgA antibodies, and so as a result of that, they can't be tagged for destruction, they can't be tagged for phagocytosis then, as a result of breaking down the IgA. And in this micrograph, you can see with adherence by the fimbriae and the enzymatic breakdown, the kind of whitish colored blobs are the bacterial cells, and you'll notice that they have these kind of dark halos around them. That's due to the enzymatic breakdown of the membrane, and leading to getting into the cell and then parasitizing the cell, getting what those bacterial cells need from the host. And again, this is group behavior activity that's going on here. Like with, as I mentioned in part one with the biofilms, where the bacteria only behave this way when they're in large groups. By forming a biofilm, they will do this as well. And if they don't realize that there's a number of them together, that they're forming a colony, they don't carry this out. They're not as pathogenic. They don't behave as pathogenically. It's like teenagers. You know, you're with one or two of them, and they're for the most part great. You get along with them just fine, and they actually behave like they're mature, and you're kind of dumbfounded by that. But then you get a group of teenagers, you get a gang of them together, say running around at, I don't know, the mall or something, and what do we could say, pathogenic. They're not quite as nice as they were all of a sudden whenever they were alone or where there was just a couple of them. Well, bacteria behave kind of the same way. When they're alone, they're quiet. Whenever they're in groups, they're ugly. And they do this kind of nonsense. Okay, major part of this particular section of this chapter would be toxins. So these are poisonous substances that contribute to their pathogenicity. This is the number one way microbes cause damage, especially bacteria. They produce fever, shock, cardiovascular issues, diarrhea, inhibit protein synthesis within the cell, and they cause all sorts of problems for us. And again, as I mentioned in part one, it's not the cells themselves that cause problems. It's the junk that they produce, the toxins that lead to all of these immune responses within us. Toxogenicity is the ability to produce a toxin. You'll notice it's got that GEN in there, and that anytime you see a word with GEN in it, gen, it's meaning the beginnings of, or to produce something. And so toxogenicity. Toxemia, that's the presence of toxin in the host's blood. Septicemia, for example, is the presence of bacteria in the blood. So emia means presence of, and usually referring to the blood. A toxoid, if a word ends in oid, it's like its prefix. So a toxoid is like a toxin. It's an inactivated toxin used in vaccines to stimulate antitoxin production. And so this is a preparation, a vaccination preparation where we use toxoids to stimulate an immune response so that we produce antitoxins, which is a group of antibodies specifically being produced against a toxin. So it's not the toxin, then you wouldn't want to give the toxin because that could be deadly in the vaccination, but a toxoid, it's like the toxin. And so it's going to stimulate an antitoxin production, and the toxoid is close enough to the real toxin that if you do encounter the real toxin, your antibodies will be there and they will be effective so that you then don't develop the disease. And an antitoxin, like I said, those are the antibodies against a toxin. So we'll look at exotoxins first. There are two categories of them, general categories, exotoxins and endotoxins. Exotoxins are proteins, and most of them are enzymes, and they're produced by some bacteria as part of their metabolism. They secrete them. So this is coming out of the bacterial cell, hence exotoxin. It's leaving, it's exiting, and that's what the prefix exo means. It's coming out of. They destroy host cell parts, or they disrupt the metabolism within the cell. And because they're enzymes, they can be used over and over and over again. And they're soluble, which means easily dissolved in blood, and they can be transported through the circulatory system and the lymphatic system quickly. Some of the most lethal substances known to man are exotoxins. So there would be, like, for example, the nasty family of bacteria called Clostridium botulinum, Clostridium tetani, Clostridium perfringens. Those produce lethal, highly lethal exotoxins that if you don't seek medical treatment, they're going to kill you. They are disease specific, which can be used diagnostically because they produce a particular series of signs and symptoms, and they're particular to that particular exotoxin. So they're disease specific, and so as a result we can use it as a diagnostic tool. So in one sense that's good because you can follow the diagnosis and then prescribe the correct treatment and hopefully get the right care so that the patient can be helped and rehabilitated. They're generally named after the cells they attack. So, for example, the Clostridium tetani toxin is a neurotoxin that affects the nervous system, hepatotoxins affecting the liver, cardiotoxins, and so on. So this very simple diagram shows where they're coming from. And so you got a simple bacterial cell, and then you have the exotoxins coming out of them. And as you see here, they're produced mostly they're by gram-positive bacteria. It's part of their growth and metabolism. Lysis can cause their release, into the surrounding medium, but they also secrete these exotoxins as well. So this chart is important because it summarizes some of the salient points about exotoxins, things that if you were a student in my class, I would definitely want you to know. So what is the source of exotoxins? Mostly gram-positive. Are they a metabolic product? Yes, they're byproducts of a growing cell. What are they made of? What's their chemistry? They're proteins. Do they cause fever? Not usually, but sometimes can, but usually not. Neutralized by an antitoxin? Yes, we produce antitoxins against them. And their LD50 is very small, and in some cases in almost infinitesimally small amount to cause death in 50% of the population. So there are three types of exotoxins that we'll look at. First is the AB toxin, and the AB stands for active and binding. So if you look at the picture, which is always helpful, we learned that in preschool and even a whole way up through college. Pictures are helpful. So we have the AB part there, and if you look at the second picture down, you can see that the B part is the binding part that is binding to the host cell membrane. There's some receptor, whatever it happens to be, on the cell membrane of the host cell, and the binding part will attach to it. It's very specific. The AB toxin then enters by endocytosis, and so the cell takes it in. And it's stimulated then to form this endocytotic structure within the cell. Then, once it gets down into the cell, the AB components separate. B leaves, and it heads out of the cell, as you can see in the last picture. And the A component alters cell function by inhibiting protein synthesis. And the theory is, or the hypothesis, I guess I should say, is that the A part interferes with translation at the ribosome. And it appears to be that way. And in some cases, it looks with certainty, and in other cases, it's still questionable and up for more research. But anyway, there's two parts, and that's why they're called AB, because of the active and binding part. And most exotoxins fall into this category. Second type would be membrane disrupting toxins. They do exactly what their name says. They disrupt the membrane and then they lyse the host cell. How do they do it? Making protein channels in the plasma membrane through the use of enzymes such as leukocytins, which are blowing apart white blood cells, and hemolysins, which are blasting apart red cells. And so as a result of that, they release then whatever happens to be inside the cell, which makes those for them nutrients available to them. They have an enzyme called phospholipase, which can break down the phospholipid bilayer of the cell. And then that, of course, then leads to the lysis of the host cell. Superantigens. Sounds impressive. Bacterial proteins that cause an intense immune response due to release of huge amounts of cytokines from T cells, which T cells are in specific immunity. Which that's a chapter that I will cover later. And as a result of this huge release of cytokines, we call that a cytokine storm. And it sounds so ominous, cytokine storm. It's one that, this is a term that is most definitely a scientific term. It's used in science, but it's also such a catchy term too. You know, that the media will just, they love to use terms like this, that we have a cytokine storm, and it's a great catch term to use in the media, for example. And of course, with what's going on right now, this is what's happening with coronavirus. There are basically two main categories of coronavirus. There's the ones that aren't so pathogenic, and they cause upper respiratory illness, and usually very mild. Doesn't this category of, we'll say, quote, nice coronaviruses, common cold, sniffs, you get the sniffles and some coughing, and and maybe not being able to, you have a little bit of problem breathing, but usually we just say it's a cold and we continue on. Well, that's not what SARS-CoV-2 is. SARS-CoV-2 is in the naughty list. So is MERS, the Middle East respiratory syndrome coronavirus. Those guys, they go deep down into the lungs. And they're the ones that cause lethal pneumonia. And so what happens is they travel so far down. This is kind of an aside just because, this is what's going on right now. Everybody's corona this and corona that. So they go deep down into the lungs, and then as a result, because of the extent of the pathogenicity and the pathogenic properties of SARS-CoV, the first round back in 2003 was called SARS-CoV-1. Now we're going through SARS-CoV-2. They create such a huge amount of inflammation, release of fluids, that this really triggers an intense immune response. And so it's a cytokine storm, which leads to these severe reactions from fever to death, nausea, vomiting, diarrhea, shock, and it'll lead to accumulation of fluid in the lungs, for example, and pneumonia in the case of corona. Another example I give are the staphylococcal toxins that cause food poisoning and toxic shock syndrome. These are superantigens, and they cause a boatload of cytokines to be released from T cells. We call that a cytokine storm. So some representative examples of exotoxins. We have the number of bacterial species there on the left, and you can see the bacterium that causes diphtheria is an AB toxin. We have pyogenes, which causes strep throat, membrane disrupting erythrogenic toxin, the botulinum bacterium that is a neurotoxin, as with its cousin there, tetani, is a neurotoxin as well. Cholera, Vibrio cholerae, is an enterotoxin, an incredible amount of diarrhea and loss of bodily fluids, and Staph aureus is a superantigen, causing a cytokine storm. The column on the right, lysogeny, that just refers to do these bacteria undergo infection by viruses, so bacteriophages. For example, strep pyogenes can undergo lysogeny, and when it's not infected, when it's kind of funny to say when the bacteria are not infected by a virus, they cause strep throat. They can then become infected by a virus and cause a host of other diseases as well. So whenever they are not infected, they produce this disease. Whenever they are infected by a bacteriophage, they can produce another disease altogether. So that's the end of part 2. I will be I'll be closing out this chapter then in part 3. And again, if you have watched my other videos, thank you for watching. I hope they have been educational and enlightening for you as well. So be sure to tune back then and look for part 3 to close out this chapter on microbial pathogenicity. Thank you for watching.